We'll be looking at the major histocompatibility complex. Okay, so our outline for the lecture is we'll look at an introduction to the MHC molecule. We'll look at the structure, function of the MHC molecule, look at the nomenclature, how it is being inherited, or the genetics. Okay, we also have to look at the clinical significance of the HLA, the role of the MHC complex or the HLA as it is being called in human, in transplantation, in transfusion therapy, and in diseases. Also, we look at how they try to cross-match or type tissues for individuals who need them. So our learning objective, we try to understand and explain what the HLA system is. What is exactly the HLA system? What is it all about? We'll have a basic understanding of the organization and inheritance of the MHC molecule. Be able to describe the clinical significance of the HLA complex. We should also be able to give an overview of pre-transplantation tests and understand their significance in clinical practice, and be able to demonstrate an understanding of the principles behind the molecular technique used in HLA typing, at least at the basic level, very basic level. So what is the major histocompatibility complex? What is the HLA complex, or as it is being called in humans? This complex is the peptide display system of the adaptive immunity. Okay, its function is to display peptide fragments of proteins, antigen, for recognition by antigen-specific T cells. It functions in presenting antigens to the body immune system, the T cells. Okay, in humans, the MHC molecule or the MHC complex is known as the human leukocyte antigen, HLA. This is because they were discovered on human leukocytes. So the HLA complex is basically a complex that presents antigen for the immune system, especially the T cells, for recognition. Okay, so it is the peptide display system of adaptive immunity, and in humans, it's called HLA because they were discovered in the human leukocytes antigen. Okay, MHC was first recognized as a genetic complex that influences the ability of an organism to accept or reject transplanted tissue. Okay, the particular set of MHC molecules a certain individual expresses determine which antigen that person T helper cells or T cytotoxics can respond to. So this complex was recognized as being the determining factor to the ability of an individual that needs organ transplant. It determines the ability of that individual to accept or reject any organ being transplanted from another person. So it is this complex that plays most of the major important role in this process of tissue transplantation. So we said that MHC molecule plays three major roles: antigen presentation, autoimmune disease plays role in autoimmune disease, and transplantation. We'll be looking at this as we go along. So the developments from the history of the HLA, how it's being discovered. There are so many scientists that are involved in the discovery of this MHC complex. But in a summary, we try to summarize some of the few key important milestones here, where we have the early work of Goa on antigen responsible for allograft rejection in mice led to the discovery of the major histocompatibility complex. The development of congenic and recombinant strains of mice by snails enabled the detailed analysis of various loci of this complex. Okay, but the foundations of the HLA complex were laid by Dorset and Roth and Rosepine in 1958. These people laid a very good foundation for especially the HLA complex in humans, as they describe the presence of alloantigens on leukocytes in human sera obtained from multiparous women or patients who had received multiple transfusions in 3 separate papers that they published. So these people were able to present to us that alloantigens are present on the leukocytes, on the leukocytes of some persons, especially women that have given birth, or patients that have received some blood transfusions, So they noticed alloantigens, antigens of white blood cells coming from other persons in their human sera, So also this discovery led to their being awarded the Nobel Prize award that they were given. Then other scientists follow subsequently in the discovery of other aspects involved in this MHC complex. So looking at the MHC molecule, the MHC molecule was named so because the molecule played a crucial role in tissue major histo, major histo from the word histocompatibility. There are three classes, but 2 major classes, as we will discuss later. So there are 3 classes of MHC molecules, but along the line, we'll be looking at only class 1 and class 2, but suffice it for now for us to classify it that the 3 classes of these MHC molecules in both human and mice. In both humans and mice, the MHC genes are arranged into regions encoding 3 classes of molecules. We have the class 1 MHC gene. These are expressed on the surface of almost all nucleated cells. Major function is presentation to T cytotoxic cells present intracellular pathogens, peptides. They present intracellular pathogens or peptides to T cytotoxic cells. Then we have the class 2 MHC genes. These are expressed primarily on antigen-presenting cells. We have our classical antigen-presenting cells, the macrophages, the dendritic cells, the B cells. Major function is presentation to T helper cells. They present extracellular pathogens. The extracellular pathogens, the peptides of extracellular pathogens to T helper cells, while the MHC class 1 molecule are presenting intracellular pathogens. The MHC class 2 molecules are presenting extracellular pathogens to T helper cells. So you must be able to distinguish these 2 facts. So the class 3 MHC genes encode for secreted products that have immune function. That is components of complement C4, C2, factor B, and TNF factor. So they encode for secreted products. So we're now going to basically focus on class 1 and class 2 MHC molecules. So classification, the chromosomal location of the MHC molecules. The MHC molecules is being housed on the short arm of chromosome number 6. You can see it from these diagrams. place that is highlighted in yellow, shaded with yellow. This is the region, the location of the MHC molecules on chromosome number 6, on the short arm of chromosome number 6. From there, we have our MHC class 2 genes, MHC class 3 genes, and MHC class 1 gene. Okay, so this is the chromosomal location of the MHC molecule or complex. The MHC region on chromosome 6 contains two sets of genes encoding the MHC class 1 and class 2 molecules. Okay, this is the genetic arrangement, the locus, the genes that make up the MHC region. The class 3 locus contain genes for complement protein and some cytokines, which we have said earlier, it encodes secretory protein. So when you look at this diagram, you'll be able to see the regions, genetic mapping for these MHC molecules. Like we said in humans, it is called the HLA complex, human leukocyte antigen. So the genes that make up the MHC class 2 are the DPDQDR. For our class 3 MHC locus, you have find it here, complement protein C4, factor B, C2. Then for and also the cytokines, TNF alpha, LT, LTB. So these are the the MHC class 3 molecules are encoding for these secretory proteins. Then we have the class 1 locus, where we have A, B, and C. So this is the diagram depicting the genes that make up each of the MHC class we have. So expression of MHC genes. So we are going to be talking about MHC class 1. It's being nucleated cells. We say that it's being expressed by all nucleated cells. That is cells that have nucleus. And you know red blood cell does not have nucleus, so it cannot, mature red blood cell anyway, does not have nucleus, so it cannot express MHC class 1. Okay, and its function is the recognition of tumor or viral infected cells by CD8. Okay, by CD8. Its function is to recognize cells in the body that have been infected by viruses, intracellular. Okay, intracellular. Okay, so we have also the class 2 molecules, the antigen presenting cells, B lymphocytes, macrophages, dendritic cells, endothelial cells, recognize infected cells by CD4. Okay, so and then the class 3 molecules, they are located on the plasma, it's a lysis of intracellular pathogens. So on one hand, we have the complex again, as we showed in the earlier diagram, the genetic regions of the gene and the gene products. Okay. So the implication of this MHC molecule is that the transfusion or transplantation of blood components bearing allogenic HLA can induce clinically significant immunological responses. All cellular components except erythrocyte express HLA, and any plasma containing products may include HLA-specific antibodies, which are potentially harmful to the recipients. Okay, so in as much as we have our classical red blood cell screening, cross-matching, when we do our normal blood grouping and cross-matching for individuals, we find out that we also have these HLA antigen, human leukocyte antigens, which are also exist in different individuals, with each individual having his own genetic makeup. So it presents problem in transplantation because of the nature, because of the properties of these HLA molecules. It tends to give some clinical barriers. Finding a match for a tissue may be quite time consuming, okay, because of the diversity, which we will see earlier, because of the properties of the HLA complex. The polymorphic nature of this complex is quite diverse. So it makes transfusion, tissue transplantation, time consuming to search for a match for an individuals. So let's look in details to MHC class 1. So this is expressed on most nucleated cells. Different cell types lead to different levels of expression. The highest expression is on lymphocytes, lowest expression on fibroblasts, muscles, liver, and neuronal cells. Cells that lack MHC1 neurons, sperm cells, certain stages, RBC cells, okay, because they are no more nucleated. Then normal cells will display self-peptides from normal turnover of self-protein and viral peptides. Each MHC can present different peptides from the same virus. MHC1 presents endogenous peptides originates from inside the cells. So MHC class 1 molecule tend to identify cells in our body that have been infected by viruses. That is, the virus has invaded inside of the cell. So the MHC class 1 molecules plays a role in the blood and the body adaptive immune system, just like we said, plays a role to identify these cells in our body that have been infected with viruses. So it's inside of them. So it is the MHC class 1 molecule that identifies these cells that have been infected, process the peptide that's processed, presents that peptide to the body immune system so that the body can eliminate such invaders. So looking at the structure of the MHC class 1 molecules, they are heterodiamers, 45 kilodaltons, okay? A chain formed with 3 proteins associated non-covalently with a 12 kDa beta microglobulin molecules. When you look at the diagram, you will see the 3 alpha domains. Alpha chain is a transmembrane glycoprotein. The alpha chain is a transmembrane glycoprotein encoded by A, B, and C regions. Okay, the 3 regions. Okay, then the beta microglobulins, beta 2 microglobulins is encoded on a highly conserved gene on a different chromosome. Okay, the beta 2 microglobulin used as a tumor marker. Okay, that is in multiple myeloma, it can serve as a tumor marker. Its increased level shows poor prognosis. The alpha 1 and alpha 2 domains interact. That is, they form a deep groove which can bind a peptide that is 8 to 10 amino acid long. So the alpha 1 and alpha 2 domain interact to give the peptide binding clefts for this molecule. The alpha 3 region is highly conserved and interacts with CD8 on TC cells and beta 2 microglobulin, okay, and beta 2 microglobulin, okay. Beta 2 microglobulin must interact with both peptides and a chain to undergo proper folding and transport to the cell surface. So this is the structure of the MHC class 1 molecule. This is the structure of the MHC class 1 molecule, okay. So the class 2 MHC class 2 molecules are only expressed constitutively by antigen-presenting cells. We have our professional APC cells, that is professional antigen-presenting cells, the B cells, the macrophages, and the dendritic cells. Also, the non-professional APC cells, where we have the epithelial cells, thymus and thyroid, the fibroblasts, glial cells, pancreatic beta cells, vascular endothelial cells. The level of expression varies at different stages of maturation or activation. MHC class 2 is not detected on pre-B cells, but is constitutively expressed on mature B cells. The MHC class 2 is upregulated on activated monocytes and macrophages. MHC contains two different polypeptide chains, the alpha and the beta chain, which we will see in this diagram, the structure of the MHC class 2, MHC, not MCH, MHC, sorry for that, MHC, not MCH, MHC class 2 structure, MHC. Okay, so this is a diagram showing the MHC class 2 molecule structure. Okay, contains two different chains. You can see the chains. You can see the chains. Okay. You can see the chains here. It has two different chains. This is one. This is the second arm of the chain. So it contains two different chains. Alpha 1 chain and beta 2 chain form the clef. You can see it here. Alpha 1 and beta 1 form the binding cleft where the antigens that it processes are being presented to the T cells, like what we see in the MHC class 1 structure, where this peptide binding groove is being formed by the alpha 1 and alpha 2, but here it's being formed by alpha 1 and beta 1 chain. Okay. Note that beta 2 is not beta 2 microglobulin here, as it is in the MHC class 1. Peptide clef can bind, the peptide clef of the MHC class 2 molecule can bind up to 13 to 18 amino acid peptides. Okay, both the alpha chain and the beta chain, are made up of two domains. Okay, the alpha 1 and beta 2 and beta 3 and beta 2 respectively. Okay, you can see the alpha chain and the beta chain. The alpha chain is made up of two domains, alpha 1, alpha 2. The beta chain is made up of two domains, beta 1 and beta 2. They are membrane-bound glycoproteins. They extend to the membrane, okay, that contains external domains, a transmembrane segment, and a cytoplasmic tail. So this please try to describe the structure of this MHC class 2 that you can see here. You have to note the domains that form the binding clefts for these molecules, the alpha 1 and alpha 2, unlike class 1 molecule where this domain is being formed by alpha 1 and alpha 2, but here it's being formed by alpha 1 and beta 1. So you note the differences between these two classes of MHC molecule, the class 1 and the class 2, the differences in their structure, the differences in their antigen presentation, the differences in the expression, tissue expressions. Okay. The MHC allele diversity. We have said that these are contained in genes, the class 1 and class 2 and class 3. Then the locus, okay, ABC for class 1 or DQ, DPR, DP for our class 2. Then allele is a two or more alternate form of a gene at a particular locus that confer alternative characters. Okay. So we're trying to talk about some of the genetic properties of this MHC molecule here. It's polymorphic because at each locus there are multiple possible allele arrangements. Okay, and then the loci constituting the MHC are highly polymorphic. They are highly polymorphic. Genes for MHC lie close together. Recombinant frequency is 0.5%. It's with rare crossing over events. So we are still looking at the genetic characteristics of these MHC molecules. The MHC allele arrangements or inheritance, we say that each set of allele is referred to as a haplotype. Individuals inherit one allele from each of the parents. Heterozygotes, different alleles at one locus, more diverse, okay? Homozygous means that the same allele at one locus. There are more likely examples include the A36 or an A36, okay? The same allele, we say they are homozygous. Different alleles, we say they are heterozygous. Okay, these alleles are co-dominantly expressed. Both maternal and paternal gene products are expressed in the same cell. So in the inheritance of this gene, the MHC complex, the inheritance for this gene, we say they are co-dominantly expressed. So every gene that is inherited from your mother, your father, you express each of them. Okay, that's give the heterozygous status of the cell. So it's not a recessive. Each gene inherited from each of the parents are being expressed. Inheriting the same allele from both parents results in a person being homozygous for that one MHC allele. MHC type is unique, like a fingerprint. It's very, very unique. To get two people that can be same is not quite an easy task. This is bad because it creates problem in transplantation, okay? Because the MHC molecules plays a crucial role in tissue or organ transplantation. That is, it determines the level of rejection of a tissue transplant. And we are now saying that the MHC type is very, very unique. It's like the fingerprints, okay? To fingerprints, getting two people that have the same fingerprint, it's not an easy task. So we say this is bad because it creates problems in transplantation to get a match, an organ match for someone. It makes it difficult because of the high polymorphic nature of this MHC complex. Okay, so it's good, it's used in paternity testing. The good side of this molecule is that it is what we investigate when we try to carry out paternity testing. It is the MHC complex that we investigate. So in that aspect, it is good because of the highly polymorphic nature of it. To get two people that are the same is difficult. It's good in that aspect of identification, okay, paternal disputes, but in the case of transplantation, getting a match for someone that needs organ transplants, it creates a huge problem. So it has its advantages. The MHC molecule has its advantages and it has its disadvantage. So the allelic forms of MHC genes, the haplotypes in humans, okay? In an outbreed population such as human, each individual is usually heterozygous at each locus. And we have different types of genotype and phenotype. A haplotype is the set of allele inherited from one parent as a set. Okay. Possible haplotypes, we have some of the examples that is here. This is how the gene is being inherited. You inherit everything you inherit from your parents, from the paternal or maternal side. You can see the progeny expressing each of them because the gene is codominant. Each of the gene that is being inherited are being expressed on the progeny. So the HLA complex, as we said earlier, is encoded on chromosome number 6. It contains more than 200 genes and extend over at least 4 million base pairs. Okay, this is just a summary of what we have been saying earlier. The HLA class 1 alpha chain molecules encoded by A, B, and C loci in humans. Okay, the beta-2 microglobulin chain encoded by a gene outside the MHC located on chromosome 15. There are also non-classical class 1 molecules, E, G, and F, that are encoded by additional gene or groups of gene within the class 1 region. Class 2 molecules are encoded by the DPDQDR regions for both the alpha chain and beta chain. For each HLA class 2 isotype, the gene encoding the alpha and beta chain are called A and B respectively. Okay, for example, HLA DQA, HLA DQB. When there is more than one gene, including non-functional gene, a number in series is added, okay, to be able to differentiate this gene. So these are some of the genetic nomenclature we try to use to differentiate these two molecules, okay, or to classify in class, trying to classify the genes. So one of the main characteristics of HLA is its extreme polymorphism, existence of different forms of this gene. Okay, among the expressed loci, the HLA has the greatest degree of polymorphism in the human genome. It exists in different forms. Okay, the number of known alleles increase constantly, as you can see from this diagram. Okay, the discovery of a number of HLA alleles has been on the increase. Has been on the increase. Class 1, class 2 has been on increase because of the high diversity, polymorphism, different forms of this gene. So this is a slide showing the nomenclature of the HLA complex, how they try to name the HLA complex. Okay, so we have the prefix here, HLA, the gene, okay, the field, then the separator, the field 1 is the allele group, okay, the allele group. The field 2 is the specific HLA protein, okay, and the field 3 is synonymous DNA substitution in coding region. Field 4, changes in non-coding region, and the suffix denote changes in expression. Okay, so whether it is being expressed, low expression, abnormal expression, and all that. So with the nomenclature, this nomenclature, someone will be able to tell the gene and all characteristics associated with it, the protein, the group, the allele group, and all the everything associated with the gene. So this is an introductory lecture. So we are doing basic introduction to the MHC complex. So some points to remember is that the gene that encodes within the MHC region, number one, that they are highly polymorphic. That is, many alternative forms of the gene, alleles, exist for each locus or antigen within the population. Many alternative forms of a gene exist within the same population, okay? They are polygenic, that is multiple genes with the same function, but with slightly different structure. This results in tremendous diversity, HLA molecule, and T-cell immunity within human population, helping them to survive epidemic disease, but also being the main immunological barrier to clinical organ transplants, helping them to survive epidemics because these proteins, because they are highly polymorphic, different forms exist, they will be able to mount immune response even to new antigens that may invade the system. So the highly polymorphic nature of the MHC molecule has its advantage, which we stated earlier. It has its advantage in paternity testing, and also helping the population to survive epidemics. That is, being able to mount immune response, being able to mount an immune response on newly evolving pathogens. Okay, it's an advantage in that area, but where it's have its disadvantage, where it's bad, the MHC molecule is bad, the angle from where it can be very, very challenging is during tissue organ transplantation, where because of the high polymorphism involved in these MHC molecules, it makes it quite a bit challenging to get a match for someone that needs to be transplanted. Take, for example, kidney. So it has its good side, it has its bad side. Tight linkage. The MHC loci are closely linked. That is, the recombination frequency within these genes is very small. Hence, alleles encoded by these loci are inherited as two sets, one from each parent. Okay, so the haplotype, each closely linked set of alleles that are inherited is called a haplotype. Codominancy, both paternal and maternal gene-derived proteins are expressed on the progeny, are expressed by the same cells, are expressed by the offspring. So there is no recessiveness of the gene of the MHC molecule. Each gene you inherit from your father, you express it. Each gene you inherit from your mother, you express it. Okay. So we may have to end this introductory lecture here because so many of you didn't join on time, so that we won't go too far and then won't go too far in this lecture. So if you have any questions you may want to ask,